Okay, happy Friday, everybody. Happy Friday. Welcome, welcome, welcome. I hope you guys had as good a week as possible. Today we're going to keep going with Unit 4 with renal physiology. We'll talk a little bit more about the nephron. As always, or as I'm structuring this, these videos won't take the full 50 minutes, so we'll go through a chunk of slides and then you'll have the rest of what would amount to class time to do those other activities for the week. Okay, so I'm gonna switch and show you Canvas very briefly. Again, if you have questions, please put them in the chat and I will get to those. So upcoming assignments, you have those 3 things that are due by midnight. So you have a homework that asks you to read the book and then fill in the chart. You have a short quiz about the comparative anatomy, and then you've got a picture I want you to draw and take a little picture of, unless you're one of those very computer savvy people that can draw things like on a tablet with a fun computer pen. What are those called? The computer pens. Whatever. If you have one of those, go for it. Otherwise, picture on your phone, submit it. Great. Next week, we have another set of lab assignments. There won't be a pre-lab quiz for next week's lab because it's really one lab broken into 3 pieces. So you'll have the next part of that renal physiology lab. You'll have another picture that I want you to draw, and then you have your final paper. Also due at the end of this unit will be those discussion questions, and they kind of span the unit. So what I recommend doing is looking at them at the beginning, like looking at them now, and keeping track in a Word document or something. And as we go through the material, just filling those out. Okay. So with that, I'm going to jump into the PowerPoint. Okay. Okay. So where we had left off, we had started to go over the nephron and the overall structure and how it's this system in your kidneys and it's full of vasculature. And what I want us to keep in mind as we dive further into detail here of the anatomy and physiology of the kidney are the functions of the kidney. So keep these in mind. The kidney needs to help us maintain the appropriate level of certain ions in our body. It needs to help us maintain osmotic balance, so the right amount of water. It needs to help us with our blood pressure. As we'll learn, the amount of water in your body relates to blood volume, which will determine blood pressure, or in part determine blood pressure. The kidney is also responsible for excreting wastes and toxins into the urine and getting them out of our body. And finally, the kidney can make its own hormones. The one I took off is the role of the kidney we're not going to really look at, which is the role of the kidney in pH balance. So we're not going to pay too much attention for this class to pH balance. So these are the 5 roles and goals that you want to keep in mind as we break down the anatomy and try to piece together what part of the kidney is helping with each of these goals. Okay. So again, last time we had briefly talked about these major processes in urine production. And these are vocab words you just need to get under your belt: filtration, reabsorption, secretion, and excretion. So filtration is going to be from the glomerulus into Bowman's capsule. This is stuff that's going to move from the blood plasma into this fluid that's in the capsule that will go through the tubule that we call the filtrate. So filtration makes our filtrate. Reabsorption then is from the tubules back into the blood, back through that capillary bed. And then secretion is the reverse of reabsorption, so things that are still in the capillaries coming into the tubules, joining the filtrate. And then excretion is everything that filtrate becomes urine and leaves the body. And again, we had shown this picture last time of the overview of where that stuff happens. So again, filtration from the glomerulus, it's that ball of capillaries through Bowman's capsule, making our filtrate. Reabsorption, things in the filtrate going back to plasma. Secretion, plasma into filtrate, and excretion, that filtrate becoming urine and leaving. Okay, let's dive down where each of these processes occur. So filtration occurs at our glomerulus. The glomerular capillary, the walls of this capillary act as a filter. Walls of the capillary act like a filter, and they're going to retain blood vessels and large molecules. So like a sieve, anything that's too big is not going to pass through. It's going to stay in the blood plasma. Everything else, based on size, is going to pass into the lumen of Bowman's capsule. So big things stay in the blood, little things move through. And you have the mesangial cells that we had talked about in this area that are going to act around the capillaries to help regulate the pressure of the filtration. So there is going to be, in the same way when you turn a hose on, if you've got a little filter in your hose, you're going to regulate the pressure, you're going to regulate the flow through the filter. So by regulating blood pressure and regulating pressure at this capsule specifically, you can control how much material passes. Okay, so that primary urine, known as filtrate, is modified by two processes, reabsorption and secretion. So the fluid that is in the lumen of the tubule, the fluid that's in our nephron tubule, we call the filtrate. You can also see it referred to sometimes as primary urine. And at the beginning, it is isoosmotic to blood. So it has the same osmolarity as blood does. And by the end, the goal of the kidney when it comes to osmotic balance is to recover 99% of the volume. So if we pass in 100 mils, we want to end up by just excreting 1 mil and taking back the other 99. And your body is going to produce about 7.5 liters of filtrate an hour, but only 75 mils of urine. So again, the vast majority of it is going to be taken back and stay in our system, and we will excrete out a small amount of water. Now, this can vary depending on your hydration status, depending on your activity level, your environment, all of that, but this is an average. The other thing to pay attention to as we move through the nephron is the epithelial wall. So again, the tubule that makes up the nephron is surrounded by one layer of epithelial tissue. But that epithelial tissue, the cells in that epithelial tissue, vary depending on where in the tubule you are. So in some, they're going to be more cuboidal, and others are going to be columnar. So the shape of the cells will change depending on where we are. And a few words that you're going to hear a lot are apical and basolateral. So the apical side faces into the lumen, into that filtrate flowing tubule. So apical into the filtrate side, whereas basolateral is into the word we call interstitium, which is the area between the tubule and the capillary. Most of the stuff that goes through the basolateral membrane of our epithelial tissue into our interstitial space is going to pass back into our capillaries. So apical into the lumen, talking about the filtrate. Basolateral, interstitial side, talking about the blood. Basolateral, blood. That's a good way to remember it. Okay. So these are some normal filtration and reabsorption rates for some typical solutes. So again, starting with water, most of it is going to be reabsorbed, 99%. So you're going to take water out of the blood, and then it's going to go back. Most of it goes back. A small amount becomes urine. Glucose is an interesting one. So in normal people, 100% of your glucose is going to be filtered and then reabsorbed. So again, anything small is going to be filtered. So the glucose is going to pass through that filter into our Bowman's capsule. But in a healthy person, your body needs glucose. And so it's like, excuse me, can I have, I need that back. And so all of the glucose that's in the filtrate is then going to pass back and eventually become back into the blood. Urea, that's the waste product that comes from the breakdown of proteins and specifically of amino acids. You reabsorb about 50% of it. So even though it's waste, about half of it is reabsorbed. Now, we'll talk about urea. It doesn't end up back in the blood for the most part. It helps create this nice concentration gradient, but that's important to keep in mind is that you're not excreting out all of your urea all the time. Sodium, we usually bring back. Chloride, we usually bring back, etc. So for most of these necessary solutes, we're going to take what we need. And these numbers, the percentage will vary depending on, again, your diet, your environment, your exertion level, all of that. Okay. So the general functions here of tubular transport. The recovery from the filtrate back into our plasma depends on a favorable gradient and on transport capacity. I'm going to say that again because it's super important. The recovery of substances from the filtrate depends on 1, a favorable gradient, and 2, transport capacity. So things need a driving force in order to move. And so that's where that gradient comes in. And they need the transporters. Most things need the transporters. Now, the exception is hydrophobic molecules. As we all know, hydrophobic molecules can just move by passive transport. So small steroids, other hydrophobic molecules, they will move from the blood into the filtrate if they're small enough, and then they'll go right back. They'll be reabsorbed passively. No need to worry about them. They'll just follow their gradient. Most molecules that we're going to talk about are not hydrophobic, and so they're going to need some sort of transport mechanism to get them from the filtrate back into the blood if they're reabsorbed. And that's going to be either through facilitated diffusion, where they fall down their concentration gradient through some sort of channel, or in some sort of active transport process. Okay, so speaking of glucose, let's talk about exactly how we reabsorb glucose. So we reabsorb glucose against its concentration gradient. So if you think of glucose, it's in your blood. It's going to, as it goes through the glomerulus, it's going to pass through that filter. It's going to be in the filtrate. All of it will be in the filtrate at the very beginning. And we need 100% of that to go back into the body. And so at some point, there's going to then be a higher concentration in the plasma than there is in the filtrate. And so you can't just let it go by passive diffusion, okay? Instead, glucose is going to use the sodium gradient. And so we're going to spend energy, our bodies are going to spend energy to make a sodium gradient, which will then provide the force that glucose can use to go against its gradient and get back into the blood. So this is showing you that right here. We've got the tubular fluid. Again, that's the filtrate. And we're going to build a concentration gradient with sodium. We're going to build a concentration gradient with sodium. And then we will use that so that glucose can fall down its concentration gradient into the peritubular space. So we're going to pump sodium out of our epithelial cells to create that force. And then the sodium glucose cotransporter will be in the epithelial tissues. And that is going to allow glucose to come down its gradient, or sodium rather, to come down its gradient into the interstitial fluid. Okay, so we use the gradient provided by sodium to move glucose. Okay, and again, in a healthy individual, 100% of that glucose is going to be reabsorbed. You're not going to see any in the ending urine. Okay, this is showing you that threshold. So renal threshold occurs when those transporters are saturated. So if we're looking at a concentration of glucose in the blood on the x-axis, and then the glucose flux, the change in glucose here on the y, you can see that for low or normal levels of glucose in your blood sugar, you're going to take all of that and it's going to be reabsorbed and it will end up in the filtrate. However, if the amount in the filtrate gets to be too high, you're going to start to see this in the urine. So if your plasma concentration gets to be too high, all of a sudden you can't reabsorb anymore. You've reached your renal threshold. All of those transporters for glucose are saturated. They're busy. They can't do any more work. And so you're going to see some appear in the filtrate and then eventually appear in the urine. And this is what happens in people who have diabetes. One of the main side effects of diabetes, or ways you can tell, is glucosuria, so glucose in the urine. And that's because all of the glucose transporters are saturated. Had enough. Okay, let's talk briefly about what we mean when we say secretion. Again, for this unit, there are some key vocab words that you need to know. So secretion is when we're going to move solutes from the blood back into the tubule. And again, this is in the opposite direction of reabsorption. And these are things like potassium, ammonia, protons, water-soluble waste products, water-soluble vitamins, things that we want to regulate the concentration of. So if we want to regulate the concentration of the blood, we might want to secrete them back into that filtrate and eventually have them excreted in the urine. Okay, each region, as we're going to talk about, each region of the nephron transports specific molecules. So this is, if you were to take the nephron and kind of stretch it out. we've got different regions. We've got the proximal tubule, we've got the descending limb of the loop of Henle, and the ascending limb of the loop of Henle, leading to our distal tubule, and a bunch of distal tubules will all come together and dump their urine out into that collecting duct. So what this is showing you is it's showing you where all of the different solutes and water transports go. So in the beginning, in that proximal tubule, we're going to reabsorb a lot of stuff right away from the filtrate. Specifically things like glucose, amino acids, some salts, going to be reabsorbed here. In the beginning, in that proximal tubule, we're also going to secrete, again, those water-soluble vitamins, ammonia, protons, they're all going to come into the proximal tubule. What you should know about the loop of Henle, and we'll spend some time going into this in more detail, is that the descending loop only moves water, and the ascending loop is where you're going to move the salts. And then in the distal tubule, again, we're going to reabsorb things like sodium and chloride and water, and we're going to secrete things like potassium, again, based on our needs. All of this is based on some key biology that we'll talk about. But again, keep in mind, things will move if there is a gradient or some sort of driving force, and if there is transport capabilities. So the specificity of solutes and water movement depends on where those transporters are. Okay. This is the same thing, but in a chart, in case you like charts. Things that are reabsorbed in the proximal tubule versus secreted, what happens in the descending versus ascending loop of Henle, the distal tubule, and the collecting duct. Okay. So overall, our proximal tubule is going to have cuboidal cells. So the cells change. In the proximal tubule, we have cuboidal cells, and these are cells that have lots of microvilli, so lots of surface area created, and lots of mitochondria to drive all of these active processes. In that first part, the descending loop of Henle, the cells are going to be cuboidal to start, and then they're going to get to a squamous cell shape, so they're going to start thick and get thin. The ascending rather goes in the opposite, so it starts thin and gets thick cells. That distal tubule, we have some principal cells, and in the collecting duct, we got principal cells and intercalated cells. And we'll break down what these cells are doing as we get to each part. Okay, so this is showing you what those cells look like. Again, we've got the proximal tubule, we've got lots of microvilli, lots of surface area. The descending limb, the cells start thick, get thin. The ascending limb, same thing. We got those thin squamous cells into those thicker cuboidal cells. And then in the collecting duct, we're going to have principal cells and intercalated cells. Okay. Let's spend a little bit of time talking about a few key ions. And the most important one is sodium. Again, because sodium is going to serve as the gradient that drives a lot of the transport processes that you see in key parts of the nephron. So we have sodium that is freely filtered. So again, that means that sodium, when we're looking at the movement across into Bowman's capsule, sodium is a small ion, so obviously it's moving across. 70% is going to be reabsorbed in the proximal tubules, distal tubules, and collecting ducts. And in general, sodium doesn't move by secretion. So once it's out of the filtrate, it's not going to come back in. And this reabsorption, as we'll learn about when we talk about hormones, it's regulated by certain key hormones to help keep that ionic balance, that key role for the kidney. And it's regulated at the end of the line. So the sodium that's moving in the proximal tubule is not under hormonal control. It's the sodium that moves at the end of the line that we can regulate. And again, we're going to use pumps to drive these processes. Okay. Potassium, also freely filtered, going to move. It's small. It's going to be reabsorbed in the proximal tubules. But unlike sodium, potassium is secreted. So in the distal tubules and the collecting ducts, potassium is going to either be reabsorbed and secreted depending on our body's needs. And it's regulated, especially in those later regions, again, by hormones and hormonal control. We'll talk about that more when we talk about hormones. Okay. With that in mind, we went over glucose, we went over sodium, we went over potassium, these big concepts I want you guys to know. We went over the cell shapes. Let's start to talk about what each part of the nephron does. So again, it depends on transport capacities and gradients. So let's break it down region by region. The proximal tubule, that first part, reabsorbs salts and organic metabolites. Most solute and water reabsorption happens here. This is where the bulk of reabsorption happens. And they're going to move from the lumen into our epithelial cells via sodium co-transporters. Things like glucose through our SGLT, lactate, amino acids, phosphates. Amino acids, you might say, like, what? Why do we need amino acids? Amino acids are nutrition. That's food, right? That's why we take in proteins. So amino acids are also going to use that sodium co-transporter because our body is like, I want these things back. I want my glucose. I want my amino acids. And then the sodium gradient is what drives us. So sodium will then be pumped into the interstitial fluid by the sodium-potassium ATPase to create that gradient. And all of this movement of salt is going to create a gradient for water. So an important concept to remember is that water is going to follow salt. You build up a lot of salt somewhere, water is going to follow. Water is going to want to fill that space. The proximal tubule also is a spot where you're going to have secretion of organic anions and cations, as well as drugs via those transporters. And again, the end result is this is where reabsorption happens for 60 to 75% of the sodium, the chloride, and the water that will eventually be reabsorbed. Okay. This is what it looks like in the proximal tubule. Again, here's the lumen with that filtrate. Here is that peritubular fluid that will eventually lead to the blood. So we're going to co-transport in sodium. The X is like, could be anything, right? Glucose, amino acids, all this stuff is going to come into these epithelial cells. And we're going to maintain a gradient where sodium will want to come into the epithelial cells by keeping the concentration low. So we keep the concentration of sodium inside the epithelial cells low with that sodium-potassium pump, okay? So keep the concentration of sodium low here so that sodium will use that gradient to come into the cell. We'll then build up a concentration of whatever X is in that epithelial cell. Again, X is moving against its gradient because sodium is the driving force. Sodium is providing the gradient. And then X will be able to fall down its gradient into the peritubular space, eventually taken up by the capillaries. And then the things that are secreted, again, those organic matter, drugs, they're going to move in the opposite direction. Okay. We're going to start the loop of Henle today as our last little section here. Loop of Henle mediates sequential uptake of water first and then salt. Water first and then salt. And the balance depends on what your body needs. So depending on your hydration levels, your exercise, all of that will determine how much water and salt is taken up. Again, the descending, this is the major site of water because transport capacities, this is where our aquaporins are at. So aquaporins are in the descending, that's going to allow water to move. And the interesting thing about the nephron is that it has an osmotic gradient. So it starts off where the proximal tubule is, where the filtrate enters. It is isoosmotic. It is equal to the osmolarity in blood, which is around 300 milliosmoles. However, as you go down, it gets saltier and saltier and saltier. And so that deep layer is going to be super salty. And it's the saltiness that allows water to move. So water is going to say, hey, I want to follow that salt when it reaches the bottom there of that descending loop. Okay. Osmotic gradient, water is going to move. Then in the ascending part, now the water can't move, just salt. And so as the osmolarity decreases, salts will then start moving. The size of the loop determines how much water is reabsorbed. So in animals, for example, that live in the desert, their nephrons have really long loops of Henle's because they want to pull as much water as possible out of their filtrate. So the longer the loop, the more water is reabsorbed. Okay, this is what that looks like. Again, up here at the top in the proximal distal tubule, we're at blood osmolarity, around 300 milliosmoles. And as we go down, as we go deeper, we get saltier and saltier and saltier. The descending moves water. So this is showing that descending, we got aquaporins, they're going to move. And the ascending moves salt. So the sodium is going to come into the epithelial cells via that sodium potassium chloride co-transporter, the NKCC, right? Moves one sodium, one potassium, two chloride into our epithelial tissue. And it's going to maintain that gradient for sodium with lots of sodium and potassium ATPases. Again, keeping the concentration in the epithelial cell low so that sodium can do the work here. Chloride will go out via a chloride potassium co-transporter or through a chloride channel. And in general, all these salts, this is where we're going to get salt reabsorption: sodium, potassium, chloride. Okay, so what we have done so far, we have started to break down the nephron piece by piece, bit by bit, talking about what happens in each of our different parts. We will continue that on Monday. We'll keep talking about what occurs in each section, and we'll build onto that by talking about how we control all of that. For the weekend, for tonight, you have those activities you have to work on, and then take a look at the discussion questions and start to think about what you would answer in each part. Okay, stop the recording.